This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 87 for June 18th, 2010. Hi everybody, it's Vincent Racaniello, and this is TWiV. And we're back to a normal TWiV with all of us in remote locations. I'm here in New York City, and returning after an absence is Alan Dove in Western Massachusetts.
Yes, I'm here, and I listened to your road trip vicariously as you and Rich were traveling about the country.
Yeah, we did a road trip. It was something. It was fun. Good to hear you again. How's everything? You well?
Yeah, things are going well.
Good.
Weather's gorgeous here now, so.
It is.
Yeah.
Moving down the coast, we have Rich Condit in North Central Florida. Hey, Rich.
Hello, Vincent, and hello to you, Alan. It's been a long time.
Yeah, been a while. Hope you're not getting hit with any tar balls down there.
Not yet. Not here in Florida, but what a mess.
Eventually. And our guest today, we actually passed him here on the East Coast on the way to Florida, is in Pittsburgh, Pennsylvania. Graham Hatfull. Hey, Graham.
Hi, how you doing? It's wonderful to be here in sunny Western PA for a change.
Graham is in the Department of Biological Sciences at the University of Pittsburgh, and you're also chair of that department. Is that right?
Yes.
I think this is the first chair we've had on TWiV, if I'm not mistaken.
Yeah, they're all too busy.
That's right. I'm glad you could make it, Graham.
Bunch of fools for the most part, I suspect.
Boy, I don't get it. You won't catch me doing that.
So, Graham, I met you in Scotland not too long ago.
Yes.
And I remember thinking you're from Pittsburgh, which is where the first polio vaccine was developed, if I'm not mistaken, right?
Yes, absolutely.
Are you well aware of that history? Yes.
It's a big part of the history here in Pittsburgh. I think that people are pleased and proud of that part of the history. A real contribution in a big way to public health.
Are you on the same campus, or is it? It was the medical school where Jonas Salk was, right?
Yes, and we're the so-called lower campus, as it's sometimes called, down in arts and sciences. But we're only about a 5-minute walk away, and we just enjoy having a very good and sort of collaborative relationships with all of the people in the School of Medicine and especially in the basic science departments here. So it's a great environment. We have Carnegie Mellon University just across the road from us as well, Duquesne University. So this is a real nice community.
Nice. And my old chairman here, Aaron Mitchell, is there at Carnegie Mellon.
He certainly is. Yes, he came just a couple of years or so ago, and he is wonderful, and I think a wonderful addition to the CMU department. A loss for you guys, I'm sorry to say.
Oh, it's a big loss. He was a big part of this department.
Yeah, really good guy, and really pleased to have him. So.
It's too bad. Well, I met you, as I said, in Scotland, and I heard you give a talk, and that's why you're here today. So I heard you give a talk, and then I gave a talk about TWiV, basically. And you said from the audience, bacteriophages are viruses too. I said, yeah, I know. Why don't you come on TWiV and talk about them? So,
and that's really terrific. It was a wonderful meeting in Edinburgh, I thought. Both the session that we talked in, which was a session with a focus on science education, but the meeting as a whole was really terrific. And I was a graduate student in Edinburgh, so it was really nice to spend a few days back there and look up a few old haunts and see a few old friends.
Oh, I didn't know that. I didn't know you were a student there. So, but you're not from Scotland, right?
No, I was born in southeast England, but and
went to graduate school. And where did you go to college?
I went to the University of London, and the specific college that I was at was called Westfield College. Real small college in North London, and I really thoroughly enjoyed it, and it's there that I first had the opportunity to get engaged in doing a little bit of investigations, doing some science, got me fired up, and I really enjoyed it.
What'd you do your PhD on?
Did my PhD with a guy called Willie Donachie on the genetics of cell division in E. coli.
And where was that?
That was in the Department of Molecular Biology at the University of Edinburgh in Scotland.
Ah, right. Okay.
Yeah.
Then that was your entree into the prokaryotic world.
Yes, I had when I was an undergraduate in London, I did a project with a guy called Peter Fay on the ultrastructure of heterocysts in Anabaena cylindrica. So that was really my initial entrance into working with prokaryotes. But then,
so what is that creature?
it's a blue-green bacterium. Used to be
Okay.
called blue-green algae. It fixes nitrogen, and it grows. Anabaena cylindrica itself grows as these filaments of cells. And if you grow under the right conditions, for example, if you deplete the availability of nitrogen, some of the vegetative cells differentiate into forming specialized cells called heterocysts. And those heterocysts are the sites of nitrogen fixation.
Excellent. Great.
Very, very cool biology. And, the nitrogenase enzymes that actually enzymatically capture or convert the nitrogen, those enzymes are very oxygen sensitive. And so that provides some rationale for why you see these specialized cells that differentiate to have that function, because a lot of the biology is set up to exclude oxygen from those cells in order to enable that process. So very cool.
You're going to love my pick of the week. It's a book I'm reading which is teaching me all this, and that's where I learned that what we used to call blue-green algae are no longer. They're cyanobacteria now, right?
Yeah, absolutely.
Very interesting. So I'm trying to trace how you got to Pittsburgh. So you were in Edinburgh, and where did you do a postdoc?
Okay, so I did what was essentially 3 postdocs, but in 2 different places. So it's a rather sort of convoluted history. When I left Edinburgh, I went and spent. So I left in 1981, and I spent about a year and a half or so working with a guy called Nigel Grinley at Yale University, working on mechanisms of recombination, and more specifically on mechanisms of transposition. And so after that, I returned to England, and I went to Cambridge to work with. Initially, I'd applied to work with Fred Sanger, and Fred retired a couple of months after I arrived there, and I worked with Bart Burrell and colleagues sequencing Epstein-Barr virus.
Wow.
And I lasted there about 15 months. I very much enjoyed myself, but was really interested in coming back to the States. Nigel was kind and generous enough, I guess, to take me back into his lab in 1984 or so, and so I spent another 4 years there working on mechanisms. Again, it's recombination mechanisms, but specifically at that point on mechanisms of site-specific recombination, these gamma delta resolvase mediated reactions. And I loved doing it. I loved being there. It was scientifically productive for me, and it was principally on the basis of those contributions that I went out looking to see if anybody would offer me a job as an assistant professor, and I applied for a position at Pittsburgh amongst other places, and Pittsburgh was one of 2 institutions that offered me a position, and I decided to come here and have been here ever since.
From the mid-'80s till now then, right?
Yeah, so I started here in 1988, and here I still am. But I've really enjoyed it here. Pittsburgh has been a wonderful community, a great place to do science, great place to pursue some of our interests in science education.
So Rich, you said there was a connection between you and Graham.
That's right. We have biography in common. One of my PhD, we've never met, but one of my PhD students, Steve Cresson, did his postdoc in Graham's lab.
So Steve was with me for a couple of years, 2 or 3 years, I don't remember exactly how long, and then moved to his own faculty position at James Madison University, where he is now. And and we continue to work with Steve and to collaborate with Steve. We just put a grant in together, and he's just maintained an interest and contributed to thinking about how we can sort of bioinformatically dissect bacteriophage genomes and understand what all the genes are and how they're related to each other. So.
And he also is quite interested in education, and that's why he chose to be at James Madison University, which is primarily an undergraduate institution rather than a research-intensive university. And I visited him there. He's doing a terrific job. As a matter of fact, the story goes on because one of the students that he had in his lab as an undergraduate is now in our graduate program. Okay.
So I believe that's true for us too.
Okay.
One of the students there one of the students that had worked there is now here as a graduate student as well. So, and I should add that JMU was, of course, Steve's alma mater as well.
Yeah, he got his dream job when he moved back there.
Yeah,
perfect for him. It's perfect for him. These, scientific families are very interesting.
Oh yeah.
It's a small world.
Well, it is.
There are lots of close-knit families, and when you dig around in your past, you can usually find that your paths have crossed somewhere.
Sure.
Well, in mathematics, that's like calculating somebody's air dose number, right?
Right. So Nigel Grinley, who was my mentor at Yale, spent time in Pittsburgh before he went to Yale, and knows some of my very close colleagues and collaborators here, like Roger Hendricks, who's we've done a lot of genomic work with bacteriophages, and so yes, those ties go back and back in interesting ways.
It's important. It's how things progress and how you maintain collaborations. It's really good. Well, I heard you talk at Edinburgh about, well, the title of your talk was "Where There's Smoke, There's Fire," PHIRE.
Yes.
And that is one of the things I wanted you to tell us about today. What is PHIRE?
PHIRE, the PHIRE program. So PHIRE is an acronym for Phage Hunters Integrating Research and Education, and it's a program that we've established here, initially going back to 2002, although it's developed quite a bit since then. And the central point, the key, the essence was really our interest in trying to find and to develop programs that can serve well to integrate our research and our educational missions. And there's, we've sort of been concerned that often research programs are constrained and restricted for students who may be gifted or tagged as academically elite or whatever, and we wanted to try to have programs that would have a much more open and open process, authentic research that essentially almost anybody could engage in, and so we've been most interested in having those programs, and I can describe the science in a minute, but we wanted programs that would be open to undergraduate students, like freshman undergraduates, or high school students, or middle school students, or school teachers, or anybody really, anybody whose principal interest was their motivation and sort of curiosity in science, as opposed to being pre-selected for having some other notion of aptitude, and so that's kind of the rationale and the reasons why we've tried to develop it. That what the science involves is the process of going out and isolating for students to go out and isolate individual new bacteriophages from the environment, to isolate them, to name them, to grow them up, purify the particles, make DNA, to sequence the complete genomes of these phages, and to annotate them, so to delve into the bioinformatics and to identify putative protein coding genes and tRNAs and other genomic features, and then to try to figure out if you can sort of tease apart the evolutionary mechanisms and the processes that would have given rise to these viruses by doing essentially comparative analysis, by taking the genome that you've isolated and characterized, and it's your new virus that you've discovered, and to compare it with other viruses and other genomes that are out there, and see if you can kind of piece together how they got to be that way.
So this is a freshman course typically?
So we actually haven't offered it as a course per se, more as an open set of research opportunities, which can be during the summer, which is obviously for students a little bit different than during the academic year when they have a lot else going on. Mm-hmm. But we've maintained sort of complete flexibility in terms of the timing. And so what drives the students to come in and do their work is not so much a schedule, but why they do it and how they do it and when they do it is arranged much more like it would be for a graduate student. Mm. They come in, do their experiments, and spend their time arranging how to best and most efficiently perform the experiments and do them in a timely way. And so we don't think of it as a course as much as a set of sort of as a community of researchers engaging in these projects.
But they do have to formally register or somehow be part of it, right? Or no, they could just wander in and out as they. Well, that wouldn't work, I guess, right?
So they. So it depends on. So there's some students that we have working in the summer, for example, where we pay them fellowships, and so they're doing a paid fellowship, and that establishes a set of of structures, if you like. But we do have quite a large number of students who essentially volunteer. They come in and participate because they want to. And they don't officially register for credit or courses or anything. They're obviously doing it with our blessing and within the administrative structures that we have set up here. But we try to keep it reasonably open so that students can take advantage of it.
What have the demographics been like so far?
So part of what's interesting about it is that we do very little advertising of this. I mean, we get large numbers of interested students. We do work quite a lot with local high schools, and so without having to do a lot of advertising, local high schools and their students are sort of aware of what's going on here, and we get lots of interest from University of Pittsburgh undergraduates, and we ask people to fill out a sort of a brief application and to make some notes as to why they're interested in doing it and some background information. But we tend not to select according to grades because we don't think that's a very reliable predictor of aptitude for doing creative scientific research, right? And so we get to sort of shape the demographic quite a lot. I mean, I can say that we clearly, we have it's gender balance. So we have about 50-50 male female. We have and have the opportunity to provide opportunities to underrepresented minorities who might not otherwise have access to such opportunities. And so, we have about 25 to 30% underrepresented minorities, which is substantially higher than you see at the University of Pittsburgh as a whole. And so we can be quite active in bringing together the demographics of the students that we want to have. And we're very pleased with the outcomes in terms of those students who have been through the program. A very large number of them stay in science. A good number go on to graduate schools, good graduate schools, and good medical schools, or other doing science in other and different ways. And we have, and again, I don't have a precise percentage, but a good number of students have co-authored papers in the peer-reviewed literature describing the genomes that they've isolated and discovered. So we see those as good indicators of success of the program.
How many students have gone through this so far, roughly?
About 60 or 70, probably. I mean, that's undergraduate students. And we have quite a lot. I mean, probably 100 or so high school students that have come in, some as volunteers and some in paid positions. we try to, as much as possible, integrate the students into the research lab, right? The idea is they're doing real, authentic research alongside our graduate students and postdocs and faculty and technicians. And so we try to avoid parceling students off into a different space, which, if we did, we could probably and conceivably introduce and bring in more students to the program. But so we draw this fine line between having it as integrated as much as possible in the research lab without completely overwhelming it.
How does that work? Do they end up interacting with students and PhD students and postdocs, or are they intimidated?
I think probably intimidated at first, but I think it wears off. One of the components that we try to introduce is that if you look at what the students are really doing in terms of the project, that there's a lot of technical and sort of structural similarity, and so we refer to this as a parallel project structure, right? And the advantage of having a parallel project structure is that you can have more students in that program, because students who have made further progress are in a great position to be mentors for other students. And so that helps to kind of break the ice a little bit, because there's an expectation that students are not only going to be able to take advantage of the opportunity to do their research, but there's an expectation that there's the responsibility to be educators, right? To be mentors and to develop as science educators, right? And it sort of puts students, I think, in a position where they have to be pretty communicative with other students, and they end up being pretty communicative with other people in the lab, postdocs and graduate students. And I find in general, for the most part, the students and the graduate students and the postdocs in the lab really enjoy having that opportunity to participate in the mentoring and the training of the students. And we have these sort of weekly notebook meetings that all the students come and present what they've been doing and talk about current issues and talk through the science. And normally there's several postdocs or graduate students from the lab that join in those and very actively participate. So
where do they get their phages from? You said they have to find a new phage in particular. Yeah. So how do they get them, and how do they know if it's new?
Great question. So they go and look essentially wherever students are inspired to go and grab some material and stick it in a tube. So that's included a variety of interesting avenues, I think. But soil, compost, that kind of stuff is normally productive, and so they go and dig around and collect stuff, and essentially do a simple extraction, just to add some buffer and filter out all the big stuff out of there, and then plate out directly to look for plaques on lawns of bacteria, and we use a particular bacterium that we're interested in called Mycobacterium smegmatis, and I can explain more about why we use that. But
I'd like to hear that.
Yeah, it's a useful choice, I think. And we focused on it for some time. But so the students take lawns of Mycobacterium smegmatis and plate out their environmental samples on it, and I wouldn't say every sample, it's 10 or 20% of samples. You normally see either 1 or 2 plaques, or occasionally you'll see 100 or 300 plaques. And so students will go and take 5 or 10 samples and plate them out. And usually in a day or two of doing that, most students can isolate a phage. So then, how do they know that it's a new isolate, right? That's a great question. I mean, there's only one way of really finding out, and it's not from looking at the viruses in the microscope and seeing a morphology, because lots of viruses have a similar morphologies. But you can only really find out if it's new and to what extent it's new by isolating the DNA and sequencing the genome and really analyzing the genome and studying it relative to other viruses. And that's a real hook, if you like, in progressing with the project, because isolating and seeing plaques on your original lawn is one thing, but stripping the virus, as it were, down to its underpants and examining its DNA and its genes is really what tells you to the extent that it's novel. And the reason in part why it works is because the diversity, I mean, certainly the number of bacteriophages in the environment is huge, and the diversity, you know, how many different types there are and how different they are to each other, which is kind of a key question that we're interested in answering. But we know that at its minimum, the diversity is massive, and the chances of finding a completely identical phage to one that's already been isolated is really slim. Right now, as we've gotten more and more phages and we've learned more and more what they look like and how they compare, we have certainly come up with examples where we have genomes that are, you know, have some polymorphisms, but no gross differences between them. But students are still out there isolating completely new genomes with sequences that are quite unlike anything else that anybody's identified, all in the same group of phages that fundamentally have the ability to infect exactly the same bacterial host, Mycobacteria and Smycomatous.
So probably if you had a few dozen students a year doing this, you could go for dozens and dozens of years and never get the same phage twice just in Pittsburgh, right?
Right. So, and you know, part of what's a really some interesting question is what does the same mean, and what does different mean, right? And one way of looking at it is, well, two viruses, you might consider two viruses the same if their sequences are 100% identical, right? Then that's not too difficult, right? But if you have two viruses whose genomes are completely different and they have few, if any, genes shared in common, then that's easy to conclude that they're different, right? But if phages vary, genomes vary by a few nucleotide differences or a few gene differences, then how does one think about that, right? But certainly we've not. I mean, I think the only case where we've had genomes that we know to be 100% identical at the nucleotide level are cases where we think that they were laboratory contamination.
So, speaking of differences and same, you've created, I believe this is your creation, a new sort of bioinformatic idea, and that is the family.
Yes.
So can you comment on that? I mean, I've
also
poured over Steve's programs looking at this, and it's a really interesting concept that I think has to do with all these comparisons, right?
Yeah, absolutely. But just before I do that, maybe I should mention that Vince, I think you just asked me about what, you know, if you got more and more, you could get dozens of these things. And in fact, just to kind of let you know where we're at, we have, so this program for phage discovery in genomics has been developed and adapted and adopted as a course. And it's been implemented at about 24 institutions. Next year, it'll be about 36 institutions. Under a program by the Science Education Alliance at HHMI, and that's the Howard Hughes Medical Institute, and all of maybe 570 freshman students this past year, and next year it's about 840 or so freshman students are all isolating and sequencing Mycobacteriophage genomes. And so, when I last looked at our database where we're trying to collate all of this, we have an entry of about 200 phages in total in there, of which we have about 150 complete genome sequences. And so that is growing to be a very substantial number of genomes, and it's going to continue to grow over the next few years.
Now, do you envision this expanding to other bacteria, not just Microbacteria?
I hope so. I mean, I think that so all the phages that in this collection, what they have in common is that they all are known to infect one particular strain of Mycobacterium smegmatis, a strain called MC²155 that was developed by my colleague Bill Jacobs at Albert Einstein College of Medicine, who names all his strain collection MC² something or other, right? So MC²155 is the smegmatis strain that we've used. And yes, I think it would be very informative to certainly use other substrains of Mycobacterium smegmatis, plus other strains of Mycobacteria, of which there's quite a lot out there, plus other strains of the group of the actinomycetes, Streptomyces, Nocardia, Cyanobacteria, and so on and so forth.
Right, and your screen is actually even a little bit more specific than that, since you're looking specifically for lytic phages.
Well, we're looking for phages that can form plaques.
Right.
Right. So some of those phages will be lytic and some will be temperate, but will be able to form lysogens. Yeah. So let me answer the question, Rich, about families.
Right.
And so.
Right. Sorry for the digression.
Yeah. No. It's a good digression, I think. Okay. So one of the things that's interesting and sort of very striking when you first start looking at these genomes, and we can construct and draw these maps where you take a genome and you identify the putative protein coding genes, and you can lay these out on a map that displays those genes and how they're arranged in a genome. And as you start comparing genomes, the thing that's most striking is that the genomes are architecturally mosaic. It's as though each genome was built of pieces, and each of those pieces, those mosaic tiles, if you like, corresponds to either a single gene, a small set of genes, and in some cases a whole block of genes, right? And so one of the most sort of interesting and sort of first questions that arises when you look at a new genome is, well, you have all these genes, and you'd kind of like to know where all those genes and where all the pieces come from and where they've evolved, how they've evolved, right? And so part of what you'd like to do, and what we would like to do, is to take each of those genes and to ask, for this particular gene, does it have any relatives? And if so, how many relatives, and in which genomes do those relatives reside? Okay, and so what we do, and it's actually it's been the computational side's been developed by Steve Creason down at JMU, and so what we do is take each of the putative protein sequences and compare everything against everything else, using both like a clustal search and also a blast search.
This is all within the phage families, is that correct?
This is all within just the Mycobacteriophages.
Okay, fine.
Yeah. And so for the number of genomes that we have now, that's, and I don't remember the exact number, 10,000 or 12,000 genes or so, and so they all get compared with each other. And then we essentially put them into boxes or into bins, right, which we call families, right, spelled PH.
PH, right.
Of course.
Everything is PH.
Pretty nerdy, isn't it? But it's
great.
It's a lot.
That's great.
And so we put them into families of related sequences. In other words, if two genes are related to each other because they meet a certain threshold value in a cluster or a blast cutoff, they get put into the same family. And so then we can take all of those and we can assort them into families. And we have a substantial number of families. It can be 40% or so that have essentially only a single member within. So it's disputable as to whether a single member of family maketh, but we have families that at current state only have a single member. We call those orfams, but they only have one member now. But as we add more genomes, we expect to find their family relatives, right? And those orfams will eventually become larger fams.
It's the Mycobacteriophage dark matter, huh, Vincent?
That's right. That's right.
Yeah.
So you find new genes regularly?
So So we find new genes, and in fact we have an abundance of new genes. At least if one defines new genes as genes that do not match using a blast cutoff, for example, a blast score, even a fairly relaxed blast cutoff, and you search the databases, publicly available databases, and you classify things as new that don't give you a significant match, then yes, I mean, and it's absolutely stunning. It's something like 80% of all of the sequence families that we have. And this is one of the advantages of putting them into families, is that you can then take a family or a representative of each family and ask whether it matches the databases.
Right. It makes it more manageable. Helps you
makes
focus
it manageable. And it eliminates that sort of redundancy of where you count one gene 20 times because you happen to have 20 genomes that have a copy of that gene, and they're all rather similar to each other, right? And so it strips out that sort of inherent redundancy or unevenness that can come from that. But when you do that, something like 80% of the families don't match. They're new genes. They don't match anything that's out there. That's pretty remarkable. I mean, there's thousands and thousands and thousands of genes for which we have basically absolutely no idea what they do.
And that's just coming from the phages that infect this one particular strain of this one particular species.
Amazing.
Yeah, and there's our colleagues that study phage numerology go out and take environmental samples and count viral particles using epifluorescence. I mean, the numbers that derive from that of phage abundance are really stunning, right? It's pretty clear, I think, that bacteriophage particles are, and they represent the most abundant forms of life on Earth, right? There's more bacteriophage particles than all other forms of life.
Yep. I think your colleague has estimated that they would, if placed end to end, stretch 200 million light years.
Isn't that a great number?
It's so much.
Yeah,
it's huge.
And so it's incredible. And so it's a huge population. It's very dynamic. Again, estimated 10 to the power 23 infections per second globally. And so it's easy to see how you could derive at this very, it's a very dynamic population, and you have very dynamic genomes that evolve from it. And with 10 to the power of 31 total particles, and with 80% of the genes or so that are completely new, I think it sounds amazing, but I think it's pretty clear that the phage population, harnesses essentially this massive reservoir of gene sequences, the things that nature has created, and that's where the vast majority of them lie, right? That's where the bulk of the stuff is that nature has created, is within the phage population, and we don't know what it does. It's absolutely amazing.
So, because you've recovered most of these from soil, does that mean are these infecting M. smegmatis in soil? Is that what you think?
So that's a really good question, and actually we don't know. The diversity of soil microbes is typically very high, and we haven't tried to sort of deconvolute what's in the various samples that we pulled out. We suspect that of all of these viruses that we've isolated, and as I said, they're pretty diverse, and we have lots of different types, that some of them may be infecting as, let's call it, their natural host in soil. Could be Mycobacterium smegmatis, but I'm not sure that it always is. And I think it may very well be other sort of quite closely related bacteria, but we don't know what they are. And sort of one of the things that makes us think that is the idea that when you look at the GC content of all of these phages, it's as though you can put them into groups, right? And so the GC content is quite varied. Spans from, I think, a low of around 56 or so percent GC up to about 69% GC. And it wouldn't surprise me if the GC content of the individual viruses fairly closely reflects that of the hosts. And so that variation in GC content is probably in some ways reflecting the GC content of whatever the various hosts are that are out there, right? And so even though we know that all of these viruses can indeed infect Mycobacterium smegmatis, we don't think that's what they're actually infecting as a sort of quote-unquote natural host in the environment.
Are there other Mycobacteria in the soil? Probably, as you said, we haven't discovered them. We don't even know what they are yet, right? Right,
that's right. I mean, I think that there's probably a dozen or so species of Mycobacteria that have been described that are saprophytes, and you can find out there easily in the environment. But it makes you wonder if there's not an equal number or a greater number of closely related strains and species that may just not be cultivatable as well, right? They just may not have been recognized.
Are you ever tempted to take one of these new genes and try and figure out what it does?
Yeah, that's been a challenge for some time, and obviously, I think from a geneticist point of view, what you'd like to be able to do to elucidate functions for some of these things is to construct a mutant, right? If you construct a mutant gene and where you've knocked the gene out, you can ask, well, is the gene required? Is it an essential gene for viral growth? And then can you figure out what it does? And so we've been thwarted for we were thwarted for 20 years or so without having a technology that would enable us to do that efficiently. And then things kind of changed a little bit over the past couple of years, principally because we've developed this recombinering technology that we can use in Mycobacteria. It's a technology that enables you to do gene replacement at reasonably high frequency, which just simplifies that whole process. And we've been able to apply those engineering principles to making recombinant phages. And
so can you make conditionally lethal mutants with this system?
So what we can do, what we've done so far is to take this and we've taken a single platform genome that we know this works well with, and just essentially march through attempting to knock out individual open reading frames, making nice, clean deletions so we don't have to worry about other effects, just whether that gene is required or not and what it does. And so about 60% of the genes that we've tried this with are completely non-essential, or at least let's say they're non-essential, so they can be removed and you can grow the particles. And so we don't have any need for conditional growth of those. Now, there are other genes that the other 40% or 35 to 40% that we either know that they are essential or we think they're essential. And not in every case, but in several cases, we've had success being able to construct the mutant using a complementation system, right?
Okay.
So we make a strain that's carrying just the gene that we're trying to delete and expresses the product of that gene. So it has the capacity to complement and to provide those functions for the growth of the mutant. And in that case, we can indeed isolate those mutants. We can propagate those mutants on this complementing strain. But then if we take that lysate of those virus particles and we infect just a wild type non-complementing strain, we can see and analyze the full impact of the mutant phenotype. And so, for example, in some cases, one particular gene we've been interested in, we can show that there's either a direct or an indirect impact on DNA replication. Because if we take the mutant and we grow it in a non-complementing strain, there's a very strong and prominent defect in the process of DNA replication.
So doing that technology, you have to do this one gene at a time, because you got to make a separate complementing host for every gene. Is there any chance of putting together some sort of like nonsense suppressing system or something in these bacteria so you'd have a general procedure?
So there's really two other things that we've been trying with sort of varying successes. We've made a series of nonsense suppressing strains for the Mycobacteria. So these are these are strains that express variant tRNAs that have suppressor capacity. And we actually did that some time ago. And in E. coli, you can isolate suppressors because you have redundancy in the tRNA population, right? And so you can get variant tRNAs while maintaining normal function. In the Mycobacteria, they have a very limited repertoire of tRNA genes, and there's not extensive redundancy. And so we engineered those strains by using phage-encoded tRNAs and essentially removing them from the phage genomes, manipulating the coding specificities so that we could use them as nonsense suppressors.
Terrific.
And they actually work pretty well, although we really haven't deployed it as fully as perhaps it should be, as we and others should do for isolating phage mutants.
Well, I guess I'm just going to have to do a sabbatical.
Yeah, absolutely.
You know?
We have certainly plenty of phages and plenty of genes for you to
Now, has anybody looked for RNA phages in Mycobacteria? It's obviously would be a little bit harder, but
that's a great question. So we've when we've done our phage isolation, we've typically tried to do it in order to maximize to the best that we can the diversity of things that we're going to isolate. So, for example, we normally avoid enrichment in the lab because that we feel might give a biased readout. And so the phages that we've looked at are essentially just things that form plaques, right? And we've certainly had our eyes and ears open for single-stranded DNA phages or RNA phages, and we haven't found anything like that. Every single one of the phages we've isolated are double-strand DNA tailed phages, and we find only 2 of the common morphologies. That amongst these, and I should add, double-strand DNA tailed bacteriophages appear to be the numerical majority of the types of phages in the biosphere, and you tend to find them of 3 major forms, which are sort of determined by their tail structures. There are things with the cyphiviridae have these long flexible tails. That's what most of our guys are. Then there's those are the cyphiviridae. Then there's the myoviridae that have contractile tails, and we have some of those. And then there's things called podoviridae, like T7, that have these little short stubby tails. And we don't have any of those. There's no podoviridae being isolated for the Mycobacteria at all. And we don't know whether that's an evolutionary constraint or whether it's really a physical constraint that perhaps they just can't, those don't work with the kinds of cell walls that the Mycobacteria have. But we have no podoviridae, and we've seen no RNA phages at all. It kind of makes you think that they ought to be out there, but we haven't been able to isolate it.
Yeah, I think if it's such an excess numbers game, then you might have trouble finding it. But if you keep looking, you probably will.
Yeah.
You know, I can't, with all of this to do, I can't imagine why you'd want to be chair and divert any of your attention to anything but doing this.
he drew the short straw, right?
All good things come to an end as well, right?
Is it a rotating chair?
Yes.
Oh,
yeah.
good for you.
Hopefully it's height adjustable too.
Yeah.
Yeah. Absolutely. I should say that we have a terrific department here, and I think that I certainly don't do my duties as chair because I'm just a sucker for punishment. I really like the department, and it gives me at least some pleasure in being able to facilitate and help out my faculty colleagues. And when I'm done, it will be somebody else's turn to do that.
Of course. So this is really amazing. I'm even more impressed than when you spoke in Scotland. Not only are you providing a great training experience for students, and you're using a safe system, which is terrific, but you're getting data. You're crowdsourcing bacteriophages, right? And can you imagine if every university in the country had a course like this, what we could discover?
Well, I remember way back before when they were just gearing up to start the Human Genome Project, there was some speaker came to Columbia and was talking about it, and some wag in the audience, might have been Aaron Mitchell, or raised his hand and said, "Well, why are you doing this way? Why don't you just have every medical student in the country sequence a gene?"
Yeah.
Yeah.
And make that a mandatory requirement for getting the medical degree.
That's a great idea.
Yes. Yeah.
That, of course, wasn't the way they did it, and it's probably good they didn't do it that way. But this sort of thing could be immensely powerful.
I think the other thing I should add is that this has been a tremendously sort of exciting and sort of stimulating. I mean, it's so the phage discovery process is so imbued with finding new genes and new phages. So it's endlessly fascinating and interesting.
Well, and for a student, it's terrific because the discovery is the whole thing, right?
That's right, and
they get to succeed.
And it happens in a time frame that they can appreciate too.
Absolutely, and I agree with all of those. Those are great points, and that's really true. And it's it's inspiring, right? I mean, it inspires all of us, but it's great that it inspires students who are at a position in their careers to make choices as to what they want to do, and this helps to useful information, right? Helps to make better choices. But I was going to say, it's not this is not really what the core of my lab does, right? And
sure, we know.
You mean there's something left over?
Well, we're interested not just in discovering these things, which is I don't mean this in a disparaging way, but it's a bit like sort of a glorified and very special form of stamp collecting, right? You get all these things and you can look at them and display them and organize them. But what's really cool is the various ways in which you can sort of exploit them and use them, right? And do interesting and fascinating things with them. And so this goes back to the question, which you asked about why we're using smegmatis, and we use it in part because one of its close relatives, Mycobacterium tuberculosis, is an incredible represents an incredible burden in terms of global health, right? And Mycobacterium tuberculosis kills more people than any other single infectious agent, right? And Mycobacterium tuberculosis is kind of it's not hard to grow, except you have to use containment, but it grows very slowly. And so there's been a really strong need to develop tools for manipulating it and understanding the genetics, but tools that you can use to make vaccine strains and to help discover new drugs and make new types of diagnostic systems, right? And phages they have all of that there in spades, right? So it's not just a matter of just having this fantastic collection of genomes, but thinking about how you can exploit it and do interesting things that would really make significant advances to an important problem in global health. And that's why we're interested in mycobacteria and mycobacteriophages. But similar arguments could be made for lots of other bacterial species, and one could imagine phage discovery programs with all sorts of hosts that could really represent significant advances to understanding those diseases.
Well, Graham, I could make the same argument for animal viruses. I think it would be great. The only problem is it gets a little less safe than just getting a bit of soil.
Yeah, and more expensive.
I mean, you could imagine going and catching mice and looking for all kinds of viruses out there in the woods. That would be great to have students do it, but it's harder. And you can't say, okay, go out in your backyard and catch a mouse and bring it in tomorrow.
This was part of the discussion I had with Steve when he revealed to me his interest in working at a primarily undergraduate institution. I encouraged him to go do phage because it's cheap, it's safe, it doesn't involve any animals or radioisotopes or anything like that. It's perfect.
Yeah, but maybe we can think of ways to make it safe to do animal viruses, because we shouldn't ignore them because it's a little bit, more dangerous to work on.
Right. Well, all of or at least a lot of the molecular biology can now be done without anything heinously dangerous.
Sure. I mean, you could take total RNA and just amplify it and sequence it. That's not bad, but getting the source. I mean, you don't find animal viruses in the soil, right? You have to have an animal.
Right.
So that's the problem.
Right.
I have a colleague here, Vince, that you probably know, Jim Peppers.
Yes, sure.
Yeah.
And Jim may be an interesting, could be a potential interesting
Yes,
TWiV guest at some point.
sure,
But
for sure.
Jim is interested in discovering what other new animal viruses might be out there, as clearly others are. But as you say, it's not a trivial exercise, and in part because of trying to advance this project forward, we're building a new BSL-3 facility here in order to be able to do some of those things, right? And so it's exactly as you say. It's you have to set yourself up to be able to do it in ways which you don't have to worry about so much with bacteriophages, provided that you choose your hosts
Right.
carefully.
No, it's a good idea. I'm going to get in touch with Jim. He'd be a great guest on the show to talk about those small double-stranded DNA viruses, SV40-like
Absolutely.
viruses, but also to talk to him about. I mean, we're interested in discovering viruses in mice, and I'd like to talk to him about that. That would be great.
You've been catching some mice there, Vince, haven't you?
I haven't started. I'm getting geared up. We have everything in place, so we're going to do it this summer.
That's really super.
But I'm sure we will find things in those animals, as you would find in any animal that you looked at.
Yeah,
right?
Absolutely.
Well, that's great. I love it. Anything else, gentlemen, before we move on?
I think that's about it.
It's a good session. I hope our listeners love it. And we're going to do a couple of emails just to give you a flavor, Graham, of the great email we get. Before we do, a word from the sponsor of this episode, Data Robotics, the maker of Drobo. Graham, have you ever heard of a Drobo?
Actually, where I heard of Drobo was on TWiV.
All right, that's the point.
But I guess I heard of something similar. I don't know if it's exactly Drobo or something related. But my son was who was interested in. My oldest son is an undergraduate at Case Western, and he's interested in computer programming and computer science. And he did a co-op, which is like a 7-month work experience, so formal work experience at a company just outside of Cleveland. And this particular company was very interested in storage and transfer of images, like medical images, and a core part of their interest in their business is with such storage capacities, and clearly it's really important to be able to safely store information, especially medical information, and of course our science information, and Drobo sounds like a great way to do that.
Well, I think for labs it's perfect with a lot of critical data, and you can get units with 4, 5, or 8 drive bays, and the point is whatever you copy is safe, so if any single or 2 drive fail, you can just put a new drive in and it will be recovered. Yeah,
I've already talked to both my lab and the folks that run our microscope suite here in the department at looking into these. I think it's a really great idea.
Yeah, if they want to buy one, they can use our product code TWiVPod and they get $75 to $500 off.
And I now have one.
Tell us about that, Alan.
Yeah, so I just got it.
Which one?
I got the Drobo FS.
It's a 5-unit drive.
It's a 5-bay system, and it's the nice thing about the FS is it's networked. So instead of plugging it directly into your computer with a USB or FireWire, which would be fine if you want to back up your computer, but I was kind of hoping to back up multiple machines and not have to leave one on. So the Drobo FS has an Ethernet jack on the back, and I plugged it into my LAN and tried to set it up, had a little difficulty, and then I read the instructions and set it up with no difficulty. So that's my recommendation. If you get a Drobo, you should probably go ahead and read the brief instructions that they provide.
Or get one's 20-year-old.
Or have your kid do it for you. So yeah, this thing now sits on my network and on my desk, and it has. I got a couple of Western Digital 2 terabyte drives, slipped those in there, and I I piled all of my important files onto it. I don't have a gigabit Ethernet hub yet. I have a 100 base T Ethernet. So it took a little while grinding away at that. Not the Drobo's fault. It is gigabit capable. It's just my network. But then, everything was on there, and it has, it's managing itself as a little RAID array that I don't have to deal with. And if I turn off my main computer, I can still access it from other computers on my LAN.
Yeah.
Just a really, really awesome. And I've got all my important stuff backed up there, and of course 2 terabytes. You can still hear an echo in there. It's massive.
Yeah.
And if I run out of space, I'll just slap a couple more drives in there.
That's great. They're very sweet.
I think I'm going to have to cave here soon.
Well, you can get some money off, as you know.
Yeah, I will look
Check
into
it
it.
out. Well, we've had quite a bit of support from Data Robotics. I think we have one more episode of support from them, and we've given away 3 units. The last one was given away to Eric, who happens to be a graduate student, a PhD student in microbiology in Birmingham, which I think was cool. That was randomly drawn from all the people who filled out the questionnaire, and so a PhD student in microbiology got it.
Excellent.
Cool.
But you can still get money off a Drobo. Use TWiV as the code, and check it out. They're really great if you have a lab with lots of data, or if you just have a lot of photographs like Dixon Despommier does. It really gives you some peace of mind. You're not going to lose your files.
Or if you're a sole proprietorship and your entire archive of work for the past 12 years is on your computer, you probably ought to back it up.
Would that be you, Alan?
Yes, that is exactly my experience.
Excellent. As usual, we're grateful for Data Robotics for their support of TWiV and the public education of science. We do appreciate it. All right, let's do a few emails. We have a couple here, which I think Graham will help us out with. The first one is directed at Alan. This is from James. I really enjoy hearing about the paths you and your guests took to enter the fields of virology and parasitology. I was wondering if Dr. Dove could talk for a few minutes about why and how he decided to transition into science journalism. Does he have any advice for PhD students who are considering making the switch? And how bad is the job market considering all the cuts media companies are making as of late? Many thanks, James in Japan. Well, I know Alan left science because I was a horrible advisor.
I was just driven out of science. That's absolutely right. It's why we've remained on such bad terms ever since. And no, I realized late in my graduate school career that I was much better at explaining my science than I was at doing it. And being in a place like Columbia, there were a lot of people around me who were really, really good at doing science, and not so many of them were as good as at explaining it. And I thought, I could continue on this PhD career path and follow the usual route and compete for grants with these people who are obviously more deserving of that for their research. Or maybe I could make a living explaining this stuff. And that's what I did, the latter. So I took an internship in the nature offices in New York and proceeded from there and set up shop as a freelancer. And that's what I've been doing ever since. Advice for PhD students who are considering making that switch now ties into my answer to your second question. I would say go on to do your postdoc. Find something to do for the next 3 to 5 years. Do not go into journalism right away. Maybe start a blog where you try to explain interesting research to a general audience. Get in some practice. Maybe build up a little bit of an audience. But wait for the dust to settle from this whole new media transition. And the reason for that is right now the old media companies who are still heavily capitalized in the old distribution systems are, kind of looks like some of them are circling the drain. And they're trying to figure out how to make the switch to the new media world. And some of them are going to make it and some of them aren't. I am still employed. I will say my income is, went down a little bit because of the recession, because that cuts into advertising budgets. But I still have clients. I still have a lot of the same clients I've had for years. But I'm in a rather narrow niche. I write mostly for scientific and trade journals. There are probably a couple dozen people in the world who are in my exact field. And I would say the whole field of journalism, and in particular science journalism, is trying to figure out what comes next. I do have faith that there is something coming next that will pay living wages to people like me. I just don't know exactly what it's going to look like, because there's enormous demand and growing demand for an audience. There's an audience out there for this stuff. There are people who want to read about science, who want to see shows about science, and they are more and more interested in not just the sort of Discovery Channel, Animals on the Serengeti type of stuff. They're actually interested in the hardcore science, if they can get some information on it that they can understand. And that's where audiences for a show like TWiV come from. That's where you get this whole podcasting revolution of science shows. But the question now is, how are people going to make a living at that? And I don't yet have an answer for you. So I would say be a postdoc, do something, find something to do in science for the next few years, and hopefully by then we will have figured out how this whole thing's going to work.
Great. Good answer. There you go, James.
Well, they're paying us a whole bunch of money for TWiV, right? We're becoming millionaires doing this, right?
Yeah.
Well, yeah, there is that, but not everybody can do TWiV.
Only so many virologists, yep. I do think that podcasts like TWiV, TWIP, and others can survive in some form going forward. Rich and I will try and figure out how to do it, and we'll let you know. We think it can be self-sustaining, but we haven't quite figured it out yet. Next one's from Marcy, and this is up Graham's alley. I love both your shows and would strongly encourage you to increase the frequency of TWIP. Here's the core here. I was volunteering in my son's second grade classroom this morning, reading a book about the human body with a student who needs extra practice reading. This student has struggled to be engaged by things he reads, but this morning he was full of questions, and this is why I'm writing to you. 2 pages of this short book were about blood and germs, and there was a photo of some red blood cells next to a photo of some, quote, germs, unquote, that I assume were rod-shaped bacteria. These photos spawned a lengthy session where the student asked many questions about germs, including, do they move? How do they move? How do they get inside our bodies? What shapes do they have? And a host of others. I drew him some examples of different shapes, paramecium, amoeba, Ebola, and told him that Paramecia swam using little hair-like things. I could not think of the word cilia at the time. And amoeba moved by oozing little feet-like things forward and then oozing the rest of itself into that foot-like projection. We also discussed more passive forms of movement, like being blown on the wind or being forcefully pushed by air, as in coughs or sneezes. The boy really wanted to know whether any germs had hair-like projections that could deploy to facilitate being carried by air, wind, like dandelion seeds. I told him that I was not aware of any, but that I knew some very smart scientists who knew a lot about microbes, and I would ask. So, are there any microbes or other parasitic things that can deploy hair-like projections to help them travel?
Ooh, that's a great question.
Yeah, it is a great question. I love the image here of this second grader asking all these questions. What a great kid, and what a great teacher,
Yeah,
and
absolutely.
Get that kid a microscope.
Yeah.
So that was exactly what I was going to say as well. It sounds
Get him
like
a microscope, give him a drop of pond water on a slide, and you won't hear from him for hours.
Yeah, and have him discover what's there to be seen. Absolutely. And
I don't have an answer to the question. Do you guys?
no, I don't. But I can't think of anything except I can't think of anything that would really represent a sort of a microbial sycamore seed, right?
Right. I wonder if, in general, microbes aren't perhaps small enough so that it's not necessary.
Yeah, I'm not sure that you'd actually get. You'd have to talk to a physicist or an aerodynamic engineer, but I'm not sure you'd get any benefit if you're a few microns across from
Right.
having hairs sticking out, because microbes already do travel across oceans. I mean, we can isolate microbes that blew over from Africa down in the Florida Panhandle.
Yeah, viruses seem to ride the air currents. In some cases, foot and mouth in Europe seems to across continents in some way, which may not be animal-borne. So it's been suggested that it might be airborne, but they don't have any kinds of projections that would do that.
Right. It's sort of interesting, though, that and we know that microbes sort of some do better than others in those types in different environments, right? And I was thinking specifically, of course, one of the things that we're interested in is tuberculosis, which is very much spread by the bacteria, right, in some type of droplets that are coughed,
right,
and transmitted to other people. And you don't find, there's not an obvious reservoir of Mycobacterium tuberculosis outside of the human population. And so it's kind of an interesting question. Perhaps it's not little projections, but it's an interesting question as to how that organism survives and does well or doesn't do in those types of environments when it's being transmitted from one person to another.
Of course, there are flagella that help them move short distances. So
yes, if they're swimming, yeah.
So that it might be interesting to point that out to this boy.
Right.
Then she asks, are there any good books with photos of microbes that you could recommend for early elementary kids? I'm hoping for a book with a picture of a microbe that simply says something like, here is a picture of the bacteria X. X is helpful, harmful to animals because it does Y and Z. X moves by flagellum and is passed from person to person by touch. More complicated explanations would also be fine, as I could translate them into simpler words, but something he could read himself would be ideal. Thanks for your help. Anybody know a book of that sort?
I think she ought to write one.
Yeah.
Yes.
I'm looking at my bookshelf right now and just seeing, I don't see one that's an obvious.
I found one at ASM Press called The Invisible ABCs. And it had some good reviews at Amazon. They says it's for 5 to 9-year-olds, and it says great illustrations. So we'll put a link to that, and maybe some of our listeners know of some examples. They could let us know about that.
And in addition, if you search Amazon.com for microscope, you'll see, look in toys and games, and you'll see a whole bunch of $50, $60 microscopes, some of which look like they might actually be pretty good optically. And I really think that would be, I remember as a child, first time I looked through a microscope, it just blew my mind.
Yep.
I think that's a terrific suggestion.
Yeah, good idea.
Do we know whereabouts Marcy is located, just in the country? Or
I don't. I have an email, but I don't know where she is, no.
I guess what we have here in Pittsburgh, but I think maybe possible to find elsewhere are, so we have really good outreach programs here. For example, we have a mobile lab that our department runs in collaboration with our CTSI friends in the School of Medicine, and part of that, and they go and visit schools, K through all the way K through 12. And part of what they have there is not they have microscopes, but they have this scope on a rope thing where you have where you can project from your scope on a rope directly onto a screen, right? And so lots of students can see at high magnification, or essentially all different parts of the natural world displayed on a screen that's very easy to see.
That's great.
I'm just thinking if there was some kind of a local university that has an outreach program close to Marcy, that might be another avenue to explore.
Or a science museum if she's lucky enough to be in a city with a good one.
Yeah, Good ideas. There you go, Marcy. All right, let's do one more. This is from Jason, who's in Australia. And he says, I thought you might like this article. You have to love the headline anyway. And it reads, Scientist infected with computer virus. Experiment to test potential problems with implants. And it was from May. A British scientist claims to have become the first human to be infected by a computer virus in an experiment he says has important implications for the future of implantable technology. Basically, what they did, this fellow's from the University of Reading, they infected a computer chip with a virus. They implanted the chip in this fellow and then transmitted the virus to a PC to prove that malware can move between human and computer.
This is actually kind of scary. I read this and I thought, my God, I'd never thought of this before, but when you consider all of the chips there are around running all of this different stuff and chips in medical devices,
Sure.
It's a pretty incredible idea.
Yep. It certainly will happen.
Yeah, and it certainly sort of helps to emphasize the just the amazing conceptual similarities between computer viruses and all of those other things that you talk about on TWiV, right? Yeah. Apart from the core structural differences of I guess silicon versus carbon, right?
Yeah.
There's not a lot that's different. You got lytic growth and latency and
yeah, it's very interesting. We do have a number of people from the IT industry who may like TWiV because of that, because there's some commonality. Now, James happens to be a medical scientist, but actually he works in a virus laboratory, and I can see him being interested in the computer-type viruses. So I would suggest, Jason, you read any of William Gibson's works. He has written about implants in people and all kinds of viruses infecting them, if you're interested in that sort of thing. All right, let's go on to a couple of picks of the week before we wrap it up, and let's start with Rich.
Let me see here. Okay, so my pick is the CDC Public Health Image Library. I discovered this some time ago. This is simply a library of photographic images of one sort or another that's maintained by the CDC on all things natural. If you go to this link on the front page, they have some featured, that's called, actually you'd like this, Graham, it's PHIL, P-H-I-L, Public Health Image Library. They've got their featured collections are influenza, natural disasters, environmental health, bioterrorism, and EM imagery. And you can click on any of these and sort through hundreds and hundreds of images, or you can search this thing. So it's fun just to look through. It's also, I think, really handy for stuff like teaching, healthcare providers. They say for photo librarians, biomedical researchers, public relations and communications. It's a fun site.
I've used this for a number of years. They initially rolled it out as a press site, and for years I've been turning to that when I need an image for a story or an editor needs an image for a story. It's a tremendously useful resource.
Yes, it really is.
And since they're government generated images, they're almost all public domain.
And here I am looking at this thing, and what pops up is a bifurcated needle giving somebody a vaccination for smallpox.
Oh, yeah. Right? It just popped up?
Yep.
Oh, yeah, there it is on the front page.
Yeah, there you go.
Pox. Very nice. Alan, what have you got?
Well, I have something humorous.
That's surprising.
Yes, I'm sure you're shocked. This is just a fun little movie. It's on YouTube. It's a Lego animation. It's done with Lego figures. And it's called Great Microbiologists. It's a history of microbiology told in 10 minutes using animated Legos.
Fabulous.
Yes, and be sure you watch what's going on in the background of some of the scenes as the narrator is talking.
This looks pretty good.
I'm not going to do it now because it'll get into our audio stream here. But I'll check that out. I love that stuff. That's great. Okay, my pick of the week is a book, and this is possibly one of the best microbiology books I have read. It's called March of the Microbes by John Ingraham. This is a book published by Harvard University Press just a few months ago. John Ingraham is a microbiologist. I think he was at UC Davis, and his specialty was fermentation. And he is retired, but he has written an incredible book on the part that microbes play in the world around us. He has in each chapter what he calls sightings. And he says, let's go to the salt flats and we'll see what kind of microbes are there. And he describes all the interactions between microbes and life. It is just amazing. So that's where I learned that there are no more blue-green algae, but they are cyanobacteria. You learn about how wine is fermented, how cheese is made. Here's the one I love. In the cow's 4 stomachs, is that right? 4 stomachs in a cow?
Gas.
There are bacteria, archaea, protozoa, and fungi, all of which have this incredibly interrelated relationship. The bacteria digest the cellulose and they produce things that other organisms grow on, and the cows eat the bacteria. And in the process, the archaea make methane. And did you know that all the cows on the earth every day excrete, or I should say, belch 20,000 tons of methane gas each day?
Wow.
And it turns out that methane is an even worse greenhouse gas than carbon dioxide. So this is a problem because we grow a lot of cows, right? Because we like our meat. And he says this is a big issue. So this is a great book. You will not be bored. You're going to learn so much because I didn't know half of this stuff. And here I call myself a microbiologist. I'm so focused on viruses. But you guys will all love it. And the listeners, get this book. You can wait till it gets into paperback if you'd like. It is just fabulous. March of the Microbes. I love it. And by the way, we put all of our book picks. We have a TWiV bookstore on Amazon. All of the picks of the last 87 episodes are there. So if you want to buy one, go there, and we get a few pennies per book. It helps keep TWiV going. So do that. Hey, Graham, you got anything you'd like to share with the audience?
Well, there's a book that I haven't read, but I'm very much looking forward to it, and so maybe I can just throw that out. It's a book that's just come out. It's called Coral Reefs in the Microbial Seas, and it's by an author called Forrest Rauer, that I think you maybe know, Vincent, or I've certainly heard Forrest discussed on TWiV, and Forrest has spent a lot of time looking at and exploring viral populations from a metagenomic perspective in the oceans in particular. And so this is called Coral Reefs in the Microbial Seas, and I'm looking forward to reading it, and I suspect that it might be a very interesting book for your readers to explore.
Great.
Excellent. Looks great.
Sounds
and
good.
Mary Yule is a co-author. Isn't she also on Small Things Considered?
That's right.
Ah, great.
Yes, she's one of the bloggers on that excellent blog, which we picked some time ago. Yeah.
Yeah.
Cool. Thank you, Graham.
Right.
Okay, that will do it for another TWiV. There are many ways you can listen to this podcast. You can go to iTunes, the Zune Marketplace, and either place you can subscribe. That way you automatically get new episodes when we post them. You can also listen with Stitcher Radio. That's a free app for your iPhone, iPod Touch, iPad, BlackBerry Palm, and Google Android device. It streams the podcast to your device, so you don't have to download it. You can get more information at bit.ly/StitchTWiV. You can always download episodes at twiv.tv, and where we also have show notes. If you like TWiV, tell other people about it. Links and reviews in iTunes are particularly helpful. TWiV is part of microbeworld.org, sciencepodcasters.org, and promednetwork.com, websites where you can find high-quality science content. As always, send us your questions and comments to twiv@twiv.tv. Graham, thank you so much for coming on. We really appreciate it.
Thank you. I really enjoyed it immensely. This was absolutely wonderful, and look forward to hearing lots more about more phage stuff on TWiV.
You aren't by any chance going to ASV, are you?
I'm afraid I'm not. There's a fantastic meeting that I'm leaving for tomorrow in Paris on viruses of prokaryotes. And
do the phage meetings still happen?
Yes. Yes,
Okay.
there's the Cold Spring Harbor phage meeting runs between Cold Spring Harbor and Wisconsin, alternating. Great meeting.
Well, perhaps we'll meet someday.
I hope so. Yes.
Well, and perhaps you'll come back someday and tell us about your phage research in your laboratory.
I'd love to.
It would be great.
Thanks.
Alan, good to have you back.
Yes, good to be back.
All right, and we will see you next week, I believe.
Sounds good.
And Rich, good to see you again.
Great to be here.
Rich, you're out next week, aren't you?
I think I'm out for 2
2
weeks.
weeks.
I'm traveling.
Have fun.
I will.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. We need a title for this, Graham. Listen to the possibilities.
We usually have a hard time coming up with something. Now we're going to have a hard time selecting amongst these.
We got Where There's Smoke, There's Fire, A Hatful of Bacteriophages, On Fire with Professor Graham Hatfull, A PHIREside Chat with Professor Graham Hatfull, and Kids Going Through a Phage.
I think that last one is great.
Yeah, I like all of those. You know, I kind of like the PHIREside Chat.
Yes, that's very good too.
I like. Did you do that one, Alan?
I did the last two, yes.
Oh, I would have thought that was Condit with a PHIREside Chat.
No, I'm not that clever. Alan's
I like Fire. Maybe it'll be a PHIREside Chat. Yeah.
Yeah, that's very good.
All right.
We've come. You know, we've enjoyed some phraseology on the around fire